Hi guys, this is Mrs. Allgood. Today we are talking about species and what defines a species and how we keep species distinct and separate instead of having two closely related species mate together to produce something new. So what you see here, this is the biological definition of a species. Okay, there's many different ways that a species can be defined, but this is the one that you need to know. So it says organisms are considered to be in the same species if they have the ability to interbreed and produce fertile offspring. We do expect that they would look similar to, but notice that that's not a key part to the definition. The key thing is that they have the ability to interbreed and produce fertile offspring. So we used the example with dogs the other day, and dogs can interbreed and produce fertile offspring because they're all the same species, they're just different breeds. Okay, so if I go too fast, feel free to pause my video. All right, so then we need to think about what prevents two closely related but different species from mating with each other. Okay, and the answer to that is reproductive isolation. Different species are going to be reproductively isolated from each other. In other words, there are mechanisms in place to discourage two closely related species from interbreeding. So what we mean by reproductive isolation is the existence of biological factors or barriers that attempt to prevent members of two different species from interbreeding and producing viable, fertile offspring. So think of it as Mother Nature discouraging two closely related species from interbreeding. It doesn't mean that it can never ever happen. It just means that usually when the two species interbreed, something goes wrong. Or there might be mechanisms in place to prevent them rather from even getting together in the first place. So we're going to be talking about reproductive isolation and reproductive barriers today. Okay, so on the front page, on the front side of this long sheet, you've got your prezygotic barriers. So prezygotic means it prevents a zygote from ever forming. Okay, and there are 5 types of prezygotic barriers. So think of it this way. The way we're preventing two closely related species from interbreeding is by preventing them from even getting together in the first place. That's kind of what we're doing on this front side of our long sheet. Okay, so the first one is temporal isolation. So when you hear the term temporal, you want to think time. Temporal has to do with time. So if you've ever heard that expression, I was in the wrong place at the wrong time, this is the wrong time component of that. So what we're trying to do is we're sort of trying to isolate the two different closely related species in some way. Here we're isolating them based on time. So what it says here is that temporal isolation is when mating or flowering occurs in different seasons or different times of day, so the two different species cannot mate. So what that could look like, that could be one species of frog mates in the spring and the other mates in the fall. And so because they're mating at different times of year, they're not going to be mating with each other. Or if we're talking about flowers, maybe one type of flower opens up during the day to be pollinated, and a different type of flower that's kind of closely related to it, a different species of flower that is, only opens up at night. Okay, so if they're opening up to be pollinated at different times of the day, then chances are they're not going to be cross-pollinating each other. Okay, so the example that we have here is with the eastern spotted skunk and the western spotted skunk. So the eastern spotted skunk breeds in late winter. The western spotted skunk breeds in the fall. And so obviously they look pretty similar just when you're looking at them. So we know that they're closely related, but they are two different species. And I know it says eastern and western, but for temporal to truly be an issue, it means that their paths may cross, but they're not going to want to mate with each other because they are used to mating at different times during the year. So temporal is all about time. The two organisms involved are mating either at different times of day if it's a flower or different times of year if it's an animal typically. Okay, our next one is habitat isolation. So an organism's habitat is where it lives. And, like that I said before, that expression, I was in the wrong place at the wrong time, this is the wrong place component. Okay, they might be living in the same general area, but they're not occupying the same habitat, so their paths don't tend to cross. And so you see the description there. This is when populations live in different habitats within the same general area and do not meet. So an example of that would be your western terrestrial garter snake and your western aquatic garter snake. So obviously one is mainly dwelling on land and one is mainly dwelling in the water. So they're not going to tend to mate with each other because they live in different habitats. So people always ask me, they say, well, what if they did? Well, they could. Okay, but we're talking about ways that nature or mechanisms that nature has put into place to kind of keep these two species separate and distinct. And so on the right-hand side, you see that there's two different species of frogs. One is primarily dwelling underground in like burrows, and one is dwelling above ground. So again, that would be habitat isolation because they are occupying different habitats even though they're in the same general area. All right, next up is gametic isolation. This one can be a little bit tougher to understand just because we haven't gotten into, reproduction in animals, reproduction in plants as much yet. But this is when two different species may mate with each other, but their gametes are unable to unite or fuse to form a zygote. So think about it this way. We talked about blood type in the last unit, and we said that there are markers on the outside of your blood cells that kind of mark your cells with whatever type of blood you have. With egg and sperm cells, they usually have species-specific markers on the outside of those cells so that only a sperm cell of the same species would be able to penetrate the egg cell and fertilize it. This tends to be much more important in organisms that are water dwellers primarily. Like you have some organisms that, the female will lay eggs and the male will release sperm into the water. Well, if that's the case, we're going to definitely need some species-specific markers on the outside of those cells to ensure that it's only gametes of that species fertilizing the egg cells of that species. So we see this with sea urchins, and then I'll explain the plant example as well. So sperm and egg cells of two different but similar species of sea urchins, so these are sea urchins down here and they're two different species, will not combine together. And again, that's usually because they're releasing those gametes into the water, so we've got to make sure that there's species-specific markers on them. In plants, it says the pollen tube will not form, so that's still considered to be gametic isolation. So think about it in this sense. Remember we said that the central part of a flower, that's the female part, is called the stigma. Okay, well the stigma is the top sticky part up here. Okay, the whole thing is called a carpel. So pollen, okay, can land on this stigma. Okay, and it's going to stick to it. So those are little pollen grains. If the two gametes are compatible, if this is pollen from the same species of plants, Then it will form a pollen tube that goes down this stalk, which is called the style, okay? And it will provide a passageway for these little sperm cells, okay? For the little sperm cells to be able to get down to the bottom here where the ovaries are, okay? So let's say that these are all that's an ovary and it contains all different sorts of ovules there. Again, to repeat that, these would be pollen grains. They land on the stigma. If the two organisms are the same species, then that will cause a pollen tube to form, and then sperm cells will be generated, and they can go down the pollen tube, which is going down the style, and they can fertilize the ovules that are down here in the ovary. Okay, so if you see anything about a pollen tube, that if it's two different species, if it's pollen from a different species of flower, it's not compatible, so it will not cause a pollen tube to form. So those plants then, those different species of flower would be showing gametic isolation. All right, behavioral isolation. Okay, so let's just do a little description first. This is with courtship rituals. So this could be breeding calls, this could be mating dances. Ways of attracting a mate are different between species. So if you might see some kind of animal that requires a specific courting ritual or mating dance in order for the male of that species to mate with them. Well, if you're of a different species, you wouldn't know that breeding call. You wouldn't know that mating dance. So it's going to keep the two separate species. It's going to keep them separate and distinct. Okay, so there is a bird on the Galapagos Islands called the blue-footed booby. I know, you can giggle now. I'm not here and I know you'll giggle. But this bird, obviously, it has blue feet, and they have a really cute courtship ritual. And so I'm going to show you the video. I don't know if the sound will play, but that's okay. Well, it might not let me play the video here, but I'll make sure you get to see the video of their little courtship ritual. So we'll figure out a way to make that happen. This is just another video that is showing the birds of paradise and their different courting or courtship rituals. And so we're going to move on to mechanical isolation, but hopefully if we don't get to watch those videos today, we can watch them tomorrow. Mechanical isolation. This is when the parts don't fit, is the easiest way to put that. So mating may be attempted, but morphological differences in their body plans between two different species prevents its successful completion. So the parts are not compatible with each other. And this can manifest itself in a couple of different ways. The first one is with snails. Okay, so it says, two closely related species of snails of the genus Bradybaena differ in the direction of their shell's spiral. So basically their shells are spiraling in two different directions, and because of that, their genitals cannot line up with each other, so mating cannot be achieved. And no, I don't have a video of that. We'll just leave that alone. Okay, in plants, the pollinator may not fit with the flower shape. So think about like a hummingbird that has a really long, thin beak. If the pollinator is not meant to pollinate that flower, then they won't fit together. Okay, whatever the proboscis, if it's an insect, whatever the insect or bird is using, it may not be able to pick up the pollen given the shape of the flower or where the pollen's located. That would also be mechanical isolation. Okay, so what we've just gone through is we've gone through pre-zygotic barriers. Those are all ways to prevent two closely related species from interbreeding before a zygote's even formed. Okay, so we talked about temporal isolation, habitat isolation, gametic isolation, mechanical isolation, and behavioral isolation. So you'll notice that all of the prezygotic barriers have in common that they end in the word isolation. So that's a good way to identify them as being a prezygotic barrier. A postzygotic barrier, this will happen after a zygote's formed, but usually something's wrong with it. And so that's going to discourage that mating between those closely related organisms in the future. So postzygotic means a zygote has been formed. Fertilization has happened, but something is typically wrong with that embryo. So first we have hybrid inviability. So all three types of postzygotic barriers are going to start with the word hybrid. Now a hybrid is what you would get if you crossed two different species together. You get a hybrid because it's not a full member of either species. Okay, so that's why these all start with hybrid. So the zygote produced by the mating of two different species is inviable. It does not survive. So we see this with two closely related species of frogs called Rana sylvatica and Rana pipiens. So what happens here is when these two similar species of frog get together, their offspring always dies. And it dies pretty early in development or shortly after birth. And so basically, if that kept happening to you, you might say, well, maybe we shouldn't be mating together because whenever we have an offspring, it doesn't survive. It's Mother Nature's way of discouraging that mating from happening. The next one, oh, this one's pretty neat. This is with geeps, okay? So sheep have 54 chromosomes and goats have 60. Most sheep-goat hybrids during development die, but some do survive. So this is Butterfly, who is a geep, okay? So just because we have things like hybrid inviability, like you see in this example, the different number of chromosomes is often what makes it be inviable. But sometimes you have a hybrid that does survive. We're just saying on the whole that mating is discouraged because most of these sheep-goat hybrids die during development. But this is Butterfly. Okay, next we're going to look at hybrid sterility. Okay, so again, notice this is a postzygotic barrier since it starts with hybrid. Hybrid zygotes reach maturity but are sterile, which means they're not going to be able to produce offspring. So the most famous example of that you should be aware of is a mule. A mule is the sterile hybrid of a male donkey, and a female horse. Okay, I think if you switch that around and you make it a female donkey and a male horse, I think it's called a hinny. Maybe called a hinny. I can't remember. But you need to know a mule. A mule is a hybrid between a male donkey and a female horse. So mules are very strong. They can be used for a lot of work around the farm, but they just can't have offspring. Okay? And there's some other cool examples of hybrid sterility out there that I want to make you aware of. There's a liger. Okay, so I think, is that what they talk about in Napoleon Dynamite maybe? A liger is the hybrid of a female tiger and a male lion. So only the males are sterile. So I think I found this last time I was teaching it. It says, knowing nothing about geeps, I would have guessed that any viable ones would be female and probably sterile. In accordance with Haldane's rule, the generalization that if only one sex of hybrids between species is either viable or fertile, that sex is almost invariably the homogametic one, the one with similar sex chromosomes. Since male mammals are the heterogametic sex and females are the homogametic sex, I'd expect geeps, if viable, to be female and to be sterile because of the evolutionary distance between the parental species. Here, for whatever reason, only the males are sterile, which means you could still have females that mate with a male lion or a male tiger. So the females apparently are not sterile. Notice that they are very large compared to the average size, I think, of a lion or tiger. They're very, very large. And of course, they're only found in captivity, not in nature, because tigers and lions tend to dwell on different continents. Lions are mainly found on the continent of Africa, and tigers are mainly found in Asia. And so you wouldn't tend to find them crossing paths much in nature. Okay? So this is me and my daughter a few summers ago. We went down to a large cat sanctuary down in Florida, and they had ligers. So I was amazed by them. And this one was very large. I know it's kind of hard to tell. So these 4 pictures, I think, are all the liger. And that one, that's the closest I could get. That's actually a tigon. So if we go back a second, a liger is the hybrid between a female tiger and a male lion. So a tigon is the opposite. It's a male tiger and a female lion. And so that's what that one is there. The rest of these are all the pictures of the liger. Other hybrids worth noting, there's zedonks. I don't know if Hollywild still has one of those. They used to have a zedonk there, a donkey-zebra cross. There's a zorse, a zebra-horse cross. There's a cama. Look how cute it is. That's a camel-llama cross. And then there's beefalo, a cross between buffalo and beef cattle. So this is still hybrid, usually examples of hybrid sterility. Even though all these different hybrids can happen, most of these are going to be sterile, or at least either the male or the female is sterile. Okay, then we have hybrid breakdown. So hybrid breakdown, what you have to pay attention with this is, if you remember back to Mendel, how we talked about the P, F1, and F2 generations. This is not affecting the F1s. This is affecting the F2s, the second generation. So this happens when the first generation of hybrids are viable and fertile, but their offspring are weak or infertile. Second generation, F2s. Okay? So, I know my picture has frogs because I think it can happen with a frog species, but this example here is with cotton species. And it says, hybrids between cotton species G. barbadense and G. hirsutum appear vigorous and fertile, but their offspring die early in development. So when you're doing your practice sheet for species, you've got to pay real careful attention to this and make sure you know what generation it's talking about. I always try and emphasize it as best I can, but you still have to pay attention to how it's worded. Okay? So that's the notes for today. We are going to watch the Cosmos video on evolution, which is really good. It's hosted by Neil deGrasse Tyson, and I feel like it kind of tackles some of those big picture evolution questions. I'm going to give you the species review sheet, but before you work on that, let me change this to red. I would finish your Hardy-Weinberg packet, because remember that you have a quiz tomorrow. So you will have to list the 5 conditions of Hardy-Weinberg, and then you will have problems to work. So please make sure you bring a calculator that is non-graphing. Okay, the species review sheet, if you have time, work on it tonight. If not, you can always work on it after your quiz tomorrow. But if you finished your Hardy-Weinberg packet, maybe you finished it last night, then work on your species review. Okay? Thanks so much. See you tomorrow. Bye.